All right.
All right, I think we are recording. So I have Dr. Shane Campbell-Staton, and this is, I'm going to start by kind of interspersing some questions from me and some questions from my students. But can you start by telling us a little bit about your journey to becoming a scientist, rather than me giving a long introduction about you?
Yeah. Oh man. So my journey to becoming a scientist. So I think my journey was probably a little bit different from most. Because I think typically when you hear, especially biologists, like talk about their journey, they always talk about, I was a kid and I was hiking or playing in a pond and I saw a thing or picked up a thing. And I yeah, that wasn't. I was raised in South Carolina, and it was like a single parent house, and so I was kind of a latchkey kid. So when I wasn't at school, I was in the house. So as a kid, my main connection to nature was actually through television. So I really got into nature through like Mutual of Omaha's Wild Kingdom and Jeff Corwin and Steve Irwin and those guys. And it was always a thing in the back of my head. I was like, oh man, isn't that cool? But again, I was a kid in the hood in South Carolina. So, I wasn't about to, I hadn't even been to a natural area in South Carolina, like, let alone like in some faraway place. But then, once I got to high school, I moved up to upstate New York and, went to this small little hippie school and, really started exploring my own academic interests. And, it was pretty late in high school where I decided that, maybe biology was something that was going to be interesting. And, so when I got into college, I decided to do, I was like, okay, well, let's major in biology. Unfortunately, by that point, I had not actually taken a science course, really. So
this is freshman at Rochester, is that right?
Yeah. This is my freshman year at University of Rochester, and so I went in and I said I want to be a biology major. They're like, okay, well, biology, chemistry, calculus, first semester. I was like, okay, let's do that then, and I got steamrolled, like really steamrolled, completely steamrolled. I had no idea what was going on. I had no idea what anyone was talking about. My first day of class, I felt like I was like 2 years late for the class.
Right.
And so my first year did not go well at all. And so that summer, I, went back home and, I talked to my mom. I was like, I don't think this is going to work out. I tried really hard and I did really poorly. And, I considered like, well, maybe I should quit and, find a job and do something that's not going to be expensive and embarrassing. And, she was like, well, you can do that, but, if you do that, you'll always have the question of like, what if I'd continued? And so I decided to continue. So that summer I actually I went back to my high school and I borrowed all the science textbooks I could get my hands on. And I spent that summer just reading all the high school science that I missed out on. And, and then I tried it again and did a little bit better. And the next semester I did a little bit better. And, I took an extra year to graduate. Well, by the time I was, I got to the end, I was working in the lab. I was doing, I had, a couple of summers of field research under my belt. And, and then luckily, I took a year off after that to study for the GREs and, and then spent my entire salary that year on graduate school applications. And, and then I ended up going to Harvard for graduate school. And that's sort of where I came into myself as an individual and as a scientist and started to hone in on the questions and systems that, now sort of define me as a scientist, I guess.
Awesome. So one thing that multiple students asked was why annals? Like how did you, and that the why that specifically, and then also as a bigger question, how do you get started with a new scientific question? How do you, how does that idea first come to you?
Yeah, that man, that's a great question. So why anoles, and how do you get started with a new so I'll answer them. I'll answer both of those things together, because I think that's probably most efficient. So basically, so the first day I showed up in um, at graduate school, I had two advisors, and one of them sort of arguably the world's expert on anoles. The other is, a major player in the world of genomics and phylogeography and trying to understand how environment shapes genes and evolution. And, I sat down and I told them explicitly I did not want to work on anoles.
Oh, really? I didn't.
Because I was in an anole lab during my undergrad, and I was like, okay, anoles are cool, whatever. I'm over that. I'm moving on to the big boy stuff, and so and I had this plan of like I wanted to study house geckos, right? Which they're um, they're extremely widespread, and I really wanted to know sort of where all these populations came from, because a lot of it is human mediated dispersal. Like some species supposedly came to America from Africa during the transatlantic slave trade, or exported, like transported in more modern times. And I was really interested in sort of tracing the pathways of this global invasion of this group of geckos. And I was really interested in sort of reading the story in the genes. And I quickly realized after doing some reading that it's really hard to read the story of a species in their genes when there is no genome, right? So the genome is kind of the reference,
Right.
Where you sort of put all these, all the patterns in context, but there was no genome. And so I went back and I talked with my advisor, Jonathan, who studies anoles, and and he was always very supportive. I was like, I don't want to work on anoles. He's like, hey, you don't have to work on anoles. It's all good. You do what you want to do. This is your time. Yeah. And and then I went back to him. I was like, Jonathan, there's no genome. Like, I don't know if I can do this. He was like, oh yeah, that sucks. You know what has a genome?
Come over to the anoles side.
Yeah. Come on over to the dark side. So I was like, okay, well, I guess maybe I should do that then. So that was sort of, that's how I got turned into Anolis.
Okay.
And, and then, in terms of like actually developing the question, right, so I know I had this tool that I wanted to use, which is the genome. And in terms of the question, it was just reading, right? I basically, I left his office and I just sat down and started reading papers, old papers, new papers, just trying to find sort of the holes, right? I mean, so that's how science moves forward. New scientists come in and they look at what's been done and it's like, well, we don't really know this thing, or we don't really know that thing. Yep. Or now we have these new techniques where we can ask these questions again at a finer scale. And so that's how I got, going. And I came across this paper that was written in the 1980s about the green anole, the species that has the genome sequence. And it talked about temperature and the fact that, some of these, some populations that live farther north, they were able to maintain function at colder temperatures than animals farther south. But it was a relatively small study. It was only 3 populations, and we didn't really know how the populations were related to each other. We didn't know how prevalent, this pattern was, because it's an extremely wide range. Basically, all of the southeastern United States, you can only extrapolate so much from three populations, spread across that entire range. So that's sort of how I got into my dissertation.
You had already decided to focus on sort of temperature and thermal tolerance even before the whole polar vortex thing.
Yeah. So the polar vortex study was actually, it was sort of a happenstance. Like my entire dissertation was on the evolution of cold tolerance in this one species, right? It's the species that comes from Cuba originally, which is like really warm and stable, but then it's migrated into Oklahoma, Tennessee, northern Mississippi, where it gets really cold in the winter. And it just struck me as a really odd environment for a subtropical animal to live in. So I spent my entire dissertation trying to understand what genes were involved, how gene flow and environment were linked. So all these questions. And then it's actually right after what was supposed to be my last field season that
Okay.
I was actually sitting at my office in Harvard in the MCZ, the Museum of Comparative Zoology, and I was on the Boston Globe website, and I was sort of looking at pictures. They had done this photo journal piece on the polar vortex storms in the South. And I was from South Carolina, so I was like, well, let's see how people back home are dealing with this. And it was like photos of people trapped in the Piggly Wiggly, and kids outside, playing, and people trying to push their cars through the snow, and big rigs stuck on the side of the highway. And one of the photos was a green anole dead in the snow. And I remember because I think it was 3 days before I had gone into my advisor's office, very triumphant. It's I have done, I've completed my field work. I am, I've been victorious, Yeah, and Jonathan was like, all right, well, get it written up and let's get you out of here. And then, 3 days later, I saw this and I went back into his office, and it's like, you remember when I said I was done collecting, but then this thing happened and I want to go back out? Yeah. And so luckily the museum supported me, and they, and my advisors supported me, and I went back out to the field and, collected more data. It just so happened that before the storm hit, I had this data on cold tolerance, and I collected this data on gene expression. And the whole plan was to sort of look at genetic response to latitude, but then this sort of chance event came through, and it was like, well, we can expand this a little bit and ask about how these processes play out on a more rapid timescale.
Well, what a cool combination of serendipity and clearly being well prepared, right? So you had been thinking about all these genes were floating around in your head, and then you're thinking about cold tolerance, and then because you're from the South, you see this thing and you're thinking about back home, and then because you have this unique combination of perspectives, this image just pops in your head, and a couple years later, science paper.
Yeah, it's crazy.
Is that too much of an oversimplification?
No, I think you're spot on. But the thing is, that's how science works a lot of the time. Right?
That's why I love that story. Yeah.
Yeah. I mean, people like to, the thing is, people like to think about science as this really clean, A to B situation. I have a hypothesis. Let's test this hypothesis.
Right.
The hypothesis is now tested and we have the answer.
Right.
But a lot of times, science just doesn't work like that. It's I have this hypothesis. Oh, it turns out I can't actually test that, but maybe I can test something that's somewhat similar. And then the result is well, I don't know what this means. Maybe. Let's look into this a little bit further.
Or I didn't know I had, but sure. Yeah.
Exactly. It's sort of this kind of meandering beast, and it has to be that way. And I think, one of the things that I think makes a successful scientist from a not so successful scientist is the ability to see where the new questions are, and to see and to recognize those opportunities, and also to have the fortitude to chase those new questions.
Yeah. So new questions. I'm gonna jump off my list a little bit. What, as you start this new position at UCLA, right?
Yeah.
What are you? Similar questions, new questions, similar, what's going to change, what's going to stay the same in terms of your research interests?
Yeah, so I think my research interests are going to stay largely the same. Those research interests are mostly trying to understand how species adapt to extreme environments. Right, a lot of what we know about the process of adaptation right now is based on relatively simple models, right? So we know things about coat color or differences in beak shape. These are relatively simple aspects of form and function that typically have a really simple genetic basis, right? And that has formed the foundation of what we know about the process of adaptation, which is great because we need that foundation. But a lot of the aspects of form and function that we're most interested in are not that simple. They're just way more complex, right? And these are things that they're governed by many, many genes that interact across different types of networks that are influenced by both environment and genetics. And this is everything from aspects of physiology that might be related to climate change, to aspects of disease, like things like diabetes and cancer. Like all of these are complex, what we call phenotypes, the aspects of form and function that are really complex and are influenced by both environments and genes. And I'm really interested in trying to understand this process, both on sort of like long time scales, like what we consider these like classical evolutionary time scales, thousands or millions of years, but then also in contemporary time scales. So a lot of, one of the big questions that I have moving into my job is how repeatable is the process of evolution, which is a classic question. All right. So, Stephen Jay Gould asked this question famously in like the early 90s, like if you replay the tape of evolution, do you get the same result? And in the certain cases, right, what we call cases of convergence or parallelism, the answer is yes at the whole organism level. But then the question is, even when the answer is yes, do you look the same because the same regulatory networks have been affected? If those same networks have been affected, is it because the same genes within those networks have been affected? If it's the same genes, is it the same mutations within those genes?
So we're going to be talking about the Lenski experiments on Thursday, where there are, in microbes and E. coli, you can, with these, I'm talking about the 12 strains.
Yeah.
We're going to be talking about exactly that, the repeatability.
Yeah. Perfect.
Cool.
Yeah. So, and I think this is a major question. And, Anolis, again, is a great system for trying to understand this question. You have each of these islands, that act as essentially a natural experiment. And, a lot of the questions about Anolis have been asked on the Greater Antilles, these like the 4 large islands of the Caribbean. But there are also these smaller islands, what we call the Lesser Antilles. There are like 7 islands that each have 1 or 2 species that also show this remarkable pattern of replicated adaptation in terms of their physiology and how they deal with variation in climate across the island. So I really want to use this system to understand how the repeatability of evolution plays out at different levels of biological organization.
Cool. Now, before you've mentioned climate change a couple times. There were a few students that were specifically interested in hearing more from you about the relevance of your work, either future or what's already been done specifically to climate change.
Yeah. So if we take this question of the repeatability of evolution and we sort of we flip the lens looking forward, right? The question, like this idea of the fact that, if we consider climate change, if we're trying to understand the biological response to climate change, how will species respond to these extremely, how will species respond to these extreme shifts in climate on such a rapid time scale? There's this inherent underpinning that evolution is itself predictable, right? And like this is still, this is still like a major question. And so I've gotten, especially since this polar vortex paper, I've gotten really into trying to understand how human-mediated shifts in the environment affect the genome of species, and how those genomic targets of selection then influence changes at the whole organism level. And so I have several projects that I can sort of talk briefly about. So right now I'm working on a project on Anolis looking at physiological and genomic adaptation to urban heat islands. Right, so urbanization causes these really extreme, warm, thermally stable microenvironments that are very different from surrounding natural areas. So we're trying to understand how these urban heat islands have affected physiological and genomic processes in anole populations in Puerto Rico. I've just put together a collaboration looking at sort of rapid selection in response to heat waves in Australia and Australian fruit bats. So these extreme heat waves have been, they've caused these massive bat die-offs. And we now have an opportunity to ask, how do these natural populations of fruit bats respond? And also sort of how long does that signature of selection last, right? If you have an extreme event like the polar vortex, you see this response. Is it just sort of a transient response? Is this something that, sort of goes back to normal after a few generations? Or is it something that's persistent?
Wave so that you can go back and resample those same populations and check. And are you hoping for sort of drought or heat wave so that you can go back to those same populations in the southern US and look for whether the signal of that polar vortex has changed the ability to deal with temperature on the other end?
Yeah, so trade-offs are definitely a huge question now, right? So we have these lineages in the green and old that survived this polar vortex, and now they're better. Now the population is better able to deal with cold. But what if a heat wave comes through, or what if a drought comes through? Those lineages that may have been better able to survive that type of event, they may be lost now because they just didn't have the genetic variants to survive a polar vortex. And when we consider aspects of climate change, right, the increase in the frequency and magnitude of all these different types of events, including like hurricanes and things like that, how does selection in response to one thing affect your ability to respond to another thing? This fruit bat project actually gives us a chance to start to ask this question because we have data from a previous set of heat waves, and it's predicted that there'll be a new set of heat waves that are coming in January, February, the Australian summer. So we can ask, when we look at events that are separated by a number of years, like, do you see the same targets of selection? Does previous selection affect how selection acts again on a population?
Do you get to go to Australia in January?
That is the plan.
Nice.
That's one of the reasons why I decided to do what I do.
Excellent. So that is sort of a not very seamless transition. There was one question that, just about your regular schedule. Not that there is any such thing as a regular schedule, but what is your day-to-day life? I know now you're doing your postdoc and you're transitioning to a new career, but what does your life look like on a day-to-day basis as a research biologist?
Yeah, so that depends a lot on the time of year.
Okay.
But I can sort of give you the broad scope of what a calendar year looks like. And this is actually one of the reasons why I love doing what I do, is because of this sort of progression. So basically, if we start I always start in the summer because that's typically the initiation of a project. And that summer is typically weeks to months of field research, right? And that's what I call game time. You strap on your boots and you get your headlamp and all your camping equipment and you head out into the green nowhere.
Yep.
And, for me, as, field biology is a major component of what I do. And, so I spend anywhere from 6 weeks to several months, traveling around, trying to find lizards, catch lizards. It's typically very long days, so typically wake up right around sunup and, have a meal, get everything, prepped for the day, head out into the field. And we're collecting and taking data in the field until noon, and take a break for another meal, let the heat of the day pass, go back out in the afternoon, and we're in data collection mode until pretty much sunset. Take a break for another meal, wait till dark comes on, and then we go out for a night herp. That's when you go out and get animals that are sleeping. So it's basically a 6 a.m. to 2 a.m. situation for 6 weeks to a couple months. It's like just long enough to get absolutely tired of it. You know, by the end of the field season, it's like, I want a shower, I want a bed, I want to watch TV, like I want civilization. And so like right when you're sick and tired of it, you know, take all that data, take all the animals, and you go into the lab. And then, you know, there's another couple of months of lab experimentation, right? Where you're in the lab working with the animals, doing acclimations, thinking about experimental designs and the limitations of those experimental designs. You know, but you're collecting data. And typically for me, this is like physiology data, doing respirometry, looking at aspects of oxygen consumption, or doing these critical thermal trials where we like heat or cool animals and we flip them onto their backs to see like when they lose their riding response.
I always wanted to see your lizard treadmill at Harvard, and I never did.
Yeah, you're gonna have to come out to UCLA to give it a gander. And then, you know, so that's about half a year between the field research and then the laboratory experimentation. And then there's all the computer work that comes along. So all the statistical analyses, and those are typically, you know, all day, every day at the computer, staring at the computer, running code and trying to figure out, you know, how do I run this statistic? Did I run this statistic right? Why do I have this error? What does this error mean? I need to Google things now. Someone help me. I hate my life. Oh, I found a result. Now I love my life. This, you know, sort of going through that process.
Like you shift seasonally from one thing going all day, every day until you're so sick of it. And then you get to shift to this other thing, and you do that all day, every day until you get sick of it, and then you shift again.
Exactly.
All right.
Yeah. And until it comes back full circle to field research again.
Cool.
Yeah.
Well, I definitely want to respect your time, but I know that I think probably the thing that my students are most interested in hearing from you is advice for undergrads. And, you know, what advice would you give yourself as an undergrad? What advice do you have for undergrads who are maybe considering maybe research biology? Maybe, I think a lot of my students are actually pre-med, but just general, you know, advice, maybe even, you know, anything from your experience as a person of color in biology that might be relevant to certain students that find.
Oh man,
that's a lot. I know. So just pick and choose whatever you want.
Yeah. So in terms of research experience for myself, if I were to run into myself as like a freshman in college, I think the biggest bit of advice I would have for myself personally is learn how to code.
Yes,
like computer science. Yeah, computer science is quickly becoming one of the foundations of biology across, like, and that's whether you're doing biomedical stuff, whether you're doing like basic research, obviously computational biology, you know, learn how to code and do it early, because you make your life a lot easier and it also opens up a lot of opportunities for you. And so that would be the advice I would give to myself. I'd also give to your students, you know, regardless of whether or not you want to be a doctor or, you know, a science educator or whether you want to do basic research, learn how to code, because you will be quite surprised really quickly like how useful it will become. And the other bit of advice I would have for your students is to get into the lab. You know, it's one thing to learn about science, right, in the classroom. It's another thing to learn how to do science, right? And again, whether or not you know, if you're thinking medicine and, you know, is your thing, you want to be a doctor or a surgeon, you know, there's something to be said about really learning the foundational principles of your field. And evolutionary biology, you know, obviously I'm biased because I'm an evolutionary biologist, but, you know, evolutionary biology is one of those foundational principles. And whether you're dealing with disease outbreaks, you know, or, you know, whether you're trying to understand, you know, whether or not you can put, you know, a replacement organ from another animal into your patient, all these are like foundational principles that are based on evolution. And, you know, this famous scientist, Theodosius Dobzhansky, once said, "Nothing in biology makes sense except in light of evolution."
Yeah.
And, and I think medicine definitely qualifies, So if you think that you want to do something biology related, biology tangential, or biology, get your butt in the lab,
Yeah.
and
I like
figure it out.
I like it.
And then the last thing you mentioned, being a scientist of color, which is certainly not a light topic, but I will try to condense. I'll say, for, being a person of color today has its challenges across the board. And science is no different. I think scientists, in the scientific community has a tendency to like to think of itself as extremely progressive and amazingly welcoming across the board. And it doesn't matter this and, it doesn't matter that. But the fact is it matters. And it, and you just look at the faculty, across pretty much any biology department across the country. I'm sure your university is the same way. It's, it's white men, and because it's historically been white men, and because of bias, the sort of this racial, ethnic, religious, gender bias, sexuality bias, there's always going to be some ignorance that you're going to have to deal with, right? And just and be prepared, right? And be prepared in the sense of, one, know that it's coming, and two, do not let it dissuade you. Because if you let it dissuade you, you do yourself a disservice and you do our field a disservice. Because if there's anything that I've learned as a scientist, is that diversity is always a good thing, right? And that's like from a biological standpoint, or from, a sociocultural standpoint, it's, you have to diversify your toolkit. Right? And the more diverse your toolkit is, the better you are at exploring potential opportunities. And not everyone will see that. And, later for them, they don't have to see it, right? But it's important for you to see it. And you to see that, one, like, you have a valuable contribution to make. And even besides that, if it's something that you want to do, like, don't let someone else decide whether or not you get to do that thing. Like, make that decision for yourself. Like, within the first week I got to Harvard, someone basically, a faculty member told me that basically the only reason I was there was because I was a Black man, and that now I had to work in order to prove that I actually deserved to be there. Which obviously threw me off. I was like, this country kid, going into Harvard. I've been there all of a week, and now all of a sudden, like, I'm Harvard's charity case. Obviously that was not true, and I busted my butt, to be successful. I've been relatively successul. I got myself a job, and now I have an opportunity to give other people opportunities. But if I had let that up,
I emailed that professor and said, "Look at me now."
Yes, in your face, in your face. So yeah, just be prepared. Prepare yourself and be resilient. Chase, chase those things that you believe are worthy of the chase.
Awesome. That's great. This has been. I'm looking at the clock because I think somehow maybe Zoom shuts us off at 40 minutes, but I don't know that for sure.
Okay.
It looks like we're still recording. But I know you're really busy, but I think that all of this has been gold. And I think my students are really going to enjoy hearing this. It's been wonderful talking to you. You're awesome. And thank you so much.
Thanks. Yeah, thank you. And if any of your students have any other questions, or they want any information about graduate school, if they think they might be interested in understanding, and pursuing like graduate studies in, in herpetology or phylogeography or climate change,
Awesome.
I'm obviously looking for graduate students,
Great.
but also if they're interested in pursuing anything else,
Yep.
reach out and I'm happy to have a conversation.
Oh yeah, I was going to ask you, what is the best way? Twitter, email. What's the best way for them to reach out if they wanted to?
Yes. So email is probably the best way right now is shane.campbellstaton@gmail.com. And
Perfect.
yeah, I look forward to hearing from anyone.
All right. Thank you so much, Shane. Really appreciate it.
Yeah, it's good seeing you.
You too. Have a great afternoon.
Thanks.
Bye.
Bye.